Excellent. Well, thank you so much for inviting me, Benjamin. And yeah, so today I'm eager to tell you all a little bit about some of the modeling work I did with my collaborator, an experimentalist named Hong Lei at Arizona. And back when Hong was at the University of Arizona in Tucson, he worked with John Hildebrand, who I think now works at the NIH, and Hong managed the Moth lab. And the moths that Hong studied were called hawk moths, which were quite large insects. So a full-grown hawk moth is about the size of maybe a peanut shell or perhaps a date. And when a hawk moth spreads its wings, it has a diameter that's about the size of the, say, palm of your hand. Now, if you study hawk moths, you'll see that they exhibit a broad repertoire of different behaviors. They fly around, they feed, they find mates, and they do all this in the rather dry terrain, the kind of expansive environments near New Mexico. And Hong and I were interested in the moth olfactory system, which is, well, loosely depicted here, roughly depicted here, because olfaction is a pretty important sensory modality for these animals. Now, if you actually look at a moth, a hawk moth, its head is shown in this diagram up here, you'll see that they have enormous eyes. And it's true that sight is very important, but they also have large feathery antennae, which they use when they can't use their eyes, which they use at night, and which are important for the animals finding food. They sip from the nectar of different flowers and finding mates because they can smell each other's pheromones and locate each other over the broad expanses of the Arizona desert. Now, the moth's antenna, those feathery antennae that sit on top of its head, they house many different kinds of odorant receptors. So these odorant receptor neurons, or they're called OSNs, there are many of them, 300,000 of them per antenna, roughly, situated on the periphery. So those feathery tufts you see on the antenna each have tens of thousands of little olfactory sensory neurons on them. And of these olfactory sensory neurons, as I said, they're more than 250,000 per antenna, they fall into only about 100 different categories based on the types of odorant or the type of molecule that they're each individually activated by. Now, when they're activated, the individual OSNs, the olfactory sensory neurons, substantially increase their firing rate. So they're neurons, so that means that they fire, they spike, they emit action potentials whenever they are sufficiently stimulated. So these olfactory sensory neurons, if they're not encountering the odor that they bind to, the odor that activates them, they don't fire very quickly. They have baseline firing rates that are very low, maybe 0 spikes per second, or maybe 1 spike per second, maybe up to 5 spikes per second in rare cases. But when they are activated, when they bind to the appropriate chemical in the environment, they send a much stronger signal. They fire more rapidly. And they send signals down the antennal nerve bundle shown, well, this is the antenna. This is the base of the antenna. The full antenna extends farther above outside the diagram, and the antennal nerve runs down the length of the antennal shaft and culminates at the bottom in this small lump of nervous tissue called the antennal lobe. It's not a particularly poetic name, but it's apt, because if you were to actually dissect a moth and take off the antenna, you would see a little lump at the base planted inside the animal's skull. That's the antennal lobe. Okay. And so the antennal nerve bundle carries the olfactory sensory neuron information down to this antennal lobe. And the antennal lobe itself houses the arbors of several different kinds of neurons, including inhibitory local neurons, as well as excitatory projection neurons, the latter of which send information to other organs in the insect brain, such as the mushroom bodies in the protocerebrum and the lateral horn, and other locations of the animal's I guess, yeah, olfactory system. Now, what's particularly interesting is that within the antennal lobe, the signals from the olfactory receptor neurons are segregated by odorant receptor type, and they serve as input to the different lumps, the different glomeruli within the antennal lobe. So if you look at the antennal lobe down here, when you look at the antennal nerve coming down the shaft of the antennal lobe, the antennal nerve fibers are separated and segregated into many, many different little bundles. Each bundle is called a glomerulus, indicated by G here. And the different olfactory sensory neurons that all are stimulated by the same kind of odorant molecule all feed into the same glomerulus. And there are roughly, on the order of 100, maybe 60 to 70 or so different glomeruli in the hawk moth. Some insects have fewer. For instance, the fly only has about 40, I think, whereas other insects have more. Some can have up to a few hundred. Okay, now let's see here. Now what I'm going to do is, yeah, here we go. So here's a different picture. This is another picture of the moth's olfactory system up here, the antennal lobe highlighted in this little gray box, and down here in figure panels A, B, and C, you can see as like a zoomed in version, an imaged version of the antennal lobe of a male moth, and the different glomeruli are shown here. And there are also a few larger glomeruli tucked away on one side in what's called the macroglomerular complex, or MGC. We'll get to that in a second. And that's again highlighted here. So as I mentioned, within the antennal lobe, the signals are segregated by odorant receptor type, and each little lump, each glomerulus, contains several local neurons, which we'll mainly think of as being inhibitory, although there is evidence for excitatory local neurons. The local neurons are called local because their processes, their dendrites, seem limited to the antennal lobe itself. They're often local, very local, only reaching to other neurons in the same glomerulus, whereas there are a handful of connections from the local neurons to other neurons in other glomeruli. And each glomerulus also contains excitatory projection neurons, which are excitatory and have projections reaching out to downstream areas of olfactory processing. Okay. Now, as indicated in the diagram, there are a few differences between the antennal lobe of male and female moths. Shown here. So this is the kind of a rough sketch of the antennal lobe of a female moth, and this is the rough sketch of an antennal lobe of a male moth. And as I pointed out earlier, one of the big differences is this macroglomerular complex. And the macroglomerular complex houses two glomeruli, the cumulus and the toroid, indicated by C and T, and they're sex-specific. So the male moths use these two glomeruli to respond to and detect female pheromones. Okay. Now, in our kind of up here, just to give you some foreshadowing, we're going to roughly imagine, and we'll talk about a model in a moment, but we're going to roughly imagine that each glomerulus comprises a few different types of inhibitory neurons, shown in blue and green, as well as excitatory projection neurons, shown in red. And we're going to assume that the local neurons and the projection neurons randomly connect to the other neurons in the same glomerulus via intraglomerular interactions, and that the local neurons also connect a little bit more sparsely, perhaps, to neurons in other glomeruli through what are called interglomerular interactions. All right. So just to summarize, the different glomeruli within the antennal lobe receive olfactory input from distinct classes of odorant receptor neurons on the antenna, and the segregation of olfactory information by odorant type gives rise to relatively complicated dynamics at the level of the antennal lobe. So, just for example, you might imagine a single odor in the environment, say a plant, or how do you say, the scent of another moth. Now, that odor would typically induce a response across a handful of olfactory classes. So the odor would be composed of a handful of different chemicals that the moth is sensitive to, say 4 different olfactory types. And a puff of the odor will strongly stimulate those 4 different classes of olfactory sensory neurons, which will in turn drive a strong response in the projection neurons and local neurons of those 4 associated glomeruli. The initially strong stimulation of the glomeruli will look like a surge in activity. Remember, from a baseline that's very low of, say, 0 to 5 hertz to 100 hertz or more. This surge of activity initially involves the projection neurons, which we'll call the PNs, those are the excitatory ones, and almost immediately afterwards, within a few milliseconds, starts to recruit and involve the local neurons, those inhibitory local neurons that were shown in green and blue. Now, if you wait a few dozen milliseconds or more, say 50 more milliseconds, this surge in activity will recruit enough activity across the local neuron population that the local neurons will blanket the directly stimulated glomeruli, as well as other adjacent glomeruli, with inhibition, lowering the firing rate within the directly stimulated glomeruli and dampening any participation or additional activity in the glomeruli that were not directly stimulated by the odor itself. So in this manner, the pattern of glomerular activation can actually change and shift over, say, 100 to 200 to 500 milliseconds, as the local neuron and projection neuron populations within the indirectly stimulated glomeruli compete for dominance. In fact, we actually believe that the glomerular activation patterns might form a combinatorial code encoding the odor identity, but I don't want to focus on that here. Instead, I want to focus on just 2 glomeruli, at least for now, the cumulus and the toroid in the macroglomerular complex. Okay, so within the cumulus and toroid, just to show, they're shown here, the cumulus is called the cumulus because it kind of looks like a big cloud, and the toroid is like a donut that wraps around the base of the cumulus and this tuft of neural processes here. So the typical scenario, if you stimulate neurons in the macroglomerular complex, is that there'll be an initial vigorous response. The response will persist for the duration of the odor, slight attenuation due to inhibition. And then if you remove the response, you'll see a prolonged period of suppression lasting about a second, maybe half a second to a second. And this period of suppression is referred to as the after hyperpolarization phase, or the AHP phase, by neurophysiologists, because it's a very apparent dynamical feature when you stimulate the antennal lobe and record from it. So just to show you what that might look like, here's a picture of, on top, rasters showing the response of a projection neuron to odor stimulation. 5 different trials are shown with, so here, each of these rows indicates a different trial. Each slash indicates a firing event, a spike from the recorded projection neuron. And the odor is presented roughly here at 0. It takes a little while for the information to get to the antenna. So the odor is presented here. There's a brief puff. And in response to that brief puff, the odor onset is at 0 and the offset is shortly afterwards. You'll see that the firing rate of the projection neuron increases. This histogram shown at the bottom here is the accumulation of all the 5 individual trials. And so you can see that the initially low background firing rate increases abruptly, but then almost as quickly starts to decrease. It decreases because the local neurons within the stimulated glomerulus start contributing to the dynamics, and they're inhibitory. Now, after the odor is removed, the persistent inhibition within the glomerulus sticks around, and that shuts down any further projection neuron activity until several hundred milliseconds, say half a second has passed. Right, so the projection neuron, when the odor is presented, the olfactory receptor neurons start to fire. The information is transmitted down the antennal nerve. The projection neuron starts to fire and then is quickly damped down, shut down by the local inhibition inside the glomerulus, but it still fires somewhat. But then when the odor is removed and the projection neuron no longer has any excitatory input coming from the antennal nerve, coming from the olfactory sensory neurons, all that's left is the inhibition. And so there's this long period, this after hyperpolarization phase following the initial excitation after the odor is removed. That's what physiologists can observe very easily. All right.
Now, sorry, I have a quick question, if you don't mind.
Please.
Yeah, what's the ratio between these projections and the inhibitory? Because if I understood well, you spoke only about inhibitory in this locally, and the nerve it is excitatory on projection neurons. So
here, so the antennal nerve impacts both the local neurons and the projection neurons inside each glomerulus with the antennal lobe. And your question is, what is the ratio between the excitatory currents and the inhibitory currents?
Yeah, I'm not confused, but I want to understand a little bit better, because you say locally there's only inhibitory and they are only excited by external, the nerve antenna. And
okay. This, I'm glad you asked this question, because this is really the meat. You're poking at the real structure here, which is that within each glomerulus, actually, the architecture is very unclear. Partly it's because the neurons are all very small, but also it's because each neuron inside each glomerulus, if you were to pick a single local neuron or a single projection neuron and stain it, you would see, well, first of all, this is an insect. So the actual soma of the cell is not in the glomerulus. The soma of the cell is sitting somewhere else. But what's important, the sort of functional part of the cell, are the diaphanous membrane, the tree-like structure, the dendrites of the cell, which are in the glomerulus and which overlap and twist and turn and come in very, very close proximity to many other dendritic arbors from many other neurons. So it's not entirely clear what connects to what inside the antennal lobe. It's very hard to disentangle the local network. From experiment, what we can conjecture is that the antennal nerve, the olfactory sensory neurons in the antennal nerve, they stimulate the projection neurons and the local neurons roughly equally, because if you measure from a projection neuron, immediately after odor presentation, its firing rate increases. Similarly, immediately after odor presentation, the local neuron firing rate increases. Now, I mentioned that the local neurons are inhibitory, and that's because many of the physiological experiments that were conducted between pairs of neurons in the antennal lobe, and it's kind of difficult to do this because you have to carefully patch clamp record 2 different neurons and hope that they connect to each other and see what happens when you stimulate one and measure from the other. When people have done that, they found that the majority of local neurons seem to be inhibitory with regards to their effect on the projection neurons, but there is evidence for excitatory local neurons. So in the network that we're going to talk about today, that I'll introduce later, we don't explicitly model excitatory local neurons because that would involve a additional neuronal population and an additional set of parameters, but one could add excitatory local neurons and it doesn't change the story very much. Alternatively, like we do in our model, we just assume that the projection neurons also have some excitatory effect on other neurons in the local glomeruli. Does that answer your question at all?
Yeah, thank you very much. Thank you.
All right, okay. So, so just to recap, right, after you present an odor, the projection neurons and the local neurons as well respond vigorously. The local neurons blanket the glomerulus as well as adjacent glomeruli to which they're connected with inhibition. And then when the odor is removed, that inhibition persists for, I don't know, 100 milliseconds, half a second or so, several hundred milliseconds, stopping any activity. Now, as you can probably guess, this after hyperpolarization phase prevents strong responses to subsequent pulses, as shown here on the bottom. So each subplot down here illustrates the kind of odor responses you might see sorted by order to the first, second, third, fourth, and fifth pulses in a sequence, 1, 2, 3, 4, and 5. And for the leftmost subplot, the sequence of pulses that were administered were separated only by 500 milliseconds. So after the first pulse, the second pulse is situated directly in the middle of the after hyperpolarization phase of the first pulse. And so you can see that the response is strongly attenuated, right? After the first pulse, you get a lot of firing events, and then the second pulse, which is reordered down here, doesn't have very many firing events at all. The third pulse has a few more, fourth pulse doesn't have many, and so forth, precisely because of this after hyperpolarization phase. In the middle subplot, the pulses are separated by a longer time period. Instead of only 500 milliseconds, the pulses are separated by a full second. This time, the after hyperpolarization phase, the AHP phase, has had a little bit of time to wane by the time the second pulse arrives, and so the responses are less muted. For the right subplot, the pulses are very far apart, 10 seconds. And that's long enough time for the after hyperpolarization phase of the first pulse to dissipate. And so when the second pulse comes, its response is almost as strong, maybe in this case even a little stronger, to the response of the first pulse. And so you see little, if any, attenuation. Okay. So far, this seems like a reasonable picture, right? The projection neurons, they respond to an odor stimulus. Oh wait, hold on. I'll post this. Is fine. The projection neurons respond to an odor stimulus, and as they do so, the local neurons also respond, and this blankets the glomerulus in a swath of inhibition that maybe lasts half a second or so. All right. So with that experimental, picture in mind, one can easily imagine a network with a relatively simple architecture: excitatory projection neurons, inhibitory local neurons, and maybe after you present an odor, the inhibitory local neurons, they spread some inhibition around. Okay, but there's a mystery here, and this is the mystery. If one were to coat the antennal lobe with a pharmacological agent called picrotoxin, a common drug, you could say, used by insect electrophysiologists, picrotoxin, sometimes called PTX, has an interesting effect on the system. So shown on top are traces from a projection neuron in a control moth antennal lobe. Each odor pulse on the bottom indicates firing. And so each odor pulse shown here, right? Yeah, so the odor pulses are shown with these little boxes. So each odor pulse induces firing, which is followed by an AHP phase. Now on the bottom, we show the traces for a projection neuron after PTX application in red. And you can see that the PTX application has reduced the spontaneous firing rates, so you see almost no spontaneous spikes, and increased the duration of the after hyperpolarization phase. which is not immediately consistent with our previous mental picture of what was going on, because PTX is actually a GABA blocker. PTX prevents the inhibitory neurotransmitter from acting. And yet, when you apply PTX to the antennal lobe, you see this very robust reduction in PN firing and an extension of the after hyperpolarization phase. So here's just a quick picture showing that if you apply this, you get robust results along the lines that I've described, and you can wash the PTX off, removing the effect. So this is a reversible effect. And this is a slide indicating more different experimental data showing that when you take a control moth, this is the picture you get. This is the picture you saw before. The after hyperpolarization phase is pronounced, but that after hyperpolarization phase can increase dramatically if you were to apply PTX. Okay, and yeah, so these are just other kind of corroborating experiments illustrating the effects. So what's going on? Okay, so this is my so often, let me just take a brief tangent here. Often when people give talks in computational neuroscience, they'll show a lot of equations and some network parameters and things like that. And at that point, usually when I watch these talks, I get a little lost, right? Because I don't really understand the details. I don't really understand what's important. And this huge swath of parameters and unconstrained information kind of washes over me, and I'm left without much of a sense for what mechanisms were important. So I thought that for this talk, even though I will show some equations and some network parameters and stuff later, I would try and focus on the mechanisms, right? Try and give you a sense intuitively for what's actually important in the network. Okay. So with that in mind, I have these little cartoons, right? So by looking at these cartoons, we can try and, for size, try on a few different network architectures, right? Try and see what might be consistent with this picture. Now, at first blush, what I described initially was a little bit like the network on the far left, the simple inhibitory circuit. In this network, the local neurons inhibit the projection neurons. But as I just mentioned, this architecture isn't consistent with the PTX experiments that I showed a moment ago. Because if you were to apply PTX to this system, the PTX would cut off, well, maybe not entirely cut off, but it would reduce the effectiveness of the local neurons. If you were to reduce the effectiveness of the local neurons, the projection neurons should fire more, which isn't what happens. So when you think about what might be at play here, one possible network mechanism is disinhibition, which is shown here in the central column. So when thinking about this network, you could imagine that perhaps there's heterogeneity across the local neurons, each of which are perhaps identical in the way that they function individually, but which are wired differently to the other neurons in the glomerulus. So an idealized example of local neuron heterogeneity is shown here with two distinct classes of local neurons. I'm imagining LN1s in green and LN2s in blue. And in this idealized example, the local neurons are not homogeneously, they're not randomly wired with the same statistics. Instead, we imagine that the local neuron 2s, the LN2s, inhibit the PNs, but are in turn inhibited by the LN1s. So in this example, we could imagine that based on different inputs, perhaps, the LN1s might have relatively high firing rates, which in turn prevent the LN2s from firing, allowing the PNs to fire. However, perhaps when you apply PTX to this network, the LN1s, their effect is reduced. And so they no longer prevent the LN2s from firing. The LN2s can fire, and now, even though their effect is also reduced by the PTX, the fact that they're firing at all would reduce the PN firing rate. Okay, so that's possible. Maybe you could entertain that hypothesis. You could imagine maybe something like that could occur, but there are two small issues with this idealized cartoon. The first issue is that we don't really have a network this simple inside a glomerulus. Instead, we have an interconnected network of local neurons and projection neurons, something more like what's shown over here in panel C. So the exact network statistics, of course, are not known because, as I mentioned earlier to Benjamin, the dendritic arbors within every glomerulus are very difficult to disentangle or even map out experimentally. So we don't actually know what the network is, but it does seem to be carefully cultivated by the development of the animal. The network isn't haphazard, but it looks haphazard to us. Okay, so there's a possibility that there's a heterogeneous collection of local neuron connectivity within each glomerulus, but we don't know exactly what it is. The second problem with this little cartoon, this little picture shown here, the second problem is that if we were to interpret this diagram as a composition of linear functions, if we were to imagine that the projection neurons had some firing rate, which was a function of the input, and the effect of the local neurons was to attenuate that by some factor, and then the effect of the local neuron 1s would be to attenuate the effect of the local neuron 2s by, again, some factor, then the diagram itself could be interpreted as a firing rate function, which involves a simple linear scaling. And then the application of PTX, which would reduce the effectiveness of inhibition by some factor, couldn't possibly cause the projection neuron firing rate to drop because the PTX would affect both the LN1s and the LN2s equally. However, it turns out that once we allow for the real nonlinearities of neuronal interactions, then PTX can indeed perform the role we just outlined across a relatively broad range of network parameters. So in summary, the mechanism that I think might be at work within the actual antennal lobe is that there's heterogeneity in the local neurons, as well as nonlinearities relating every neuron's firing rate to its effect on its downstream targets within the glomerulus. And that combination of heterogeneity within the local architecture and nonlinearity in terms of the link between activity and the effect of that activity can, or almost generically, produces the kind of effect that we've seen in the PTX experiments I showed earlier. So we had this experimental conundrum, mystery solved, right? It seems like one could, with this principle in mind, wire up a network and tune it and, set up a network model which can do all of these things. But wait, hold on. There's something that's more complicated. There's a wrench in the works, okay? Because it turns out that there's another GABA blocker, bicuculline, sometimes referred to as BIC, which actually has the opposite effect of PTX. If you apply bicuculline to the antennal lobe, instead of reducing the PN activity and increasing the AHP phase, what bicuculline does is it actually increases the PN activity and reduces the AHP phase. One example is shown here in black. We show the PN trace before on the left, during in the middle, and after on the right, BIC application, bicuculline application. And on the bottom, we show a control, PN, blue on top, contrasted with the BIC affected PN, red, on the bottom. And so, here, let's just, yeah, okay. So given what I've told you so far, right, there's an inconsistency. There's no possible way these two experiments could be explained with the same architecture, because so far as what you know, if PTX and BIC are each just GABA blockers, then their effect on the antennal lobe should be the same, right? But experimentally, we see that actually their effects are almost exactly opposite to one another, or very, very different. So what could be happening here? Well, it turns out that bicuculline, BIC, does quite a few things that PTX doesn't do. And after some poking around, Hong, my experimentalist collaborator, he discovered that BIC actually serves as a blocker for certain channels that PTX doesn't block. And these channels are what are called SK channels, or slow-acting calcium-dependent potassium channels. These SK channels are intrinsic channels, which themselves confer an inhibitory current to each PN, and which are affected by calcium, which is driven by that PN's own spiking activity. So just to give you a mental picture of what these SK channels do, they're ion channels that sit on the membrane of each projection neuron. And when the projection neuron itself fires many times, the calcium influx into that projection neuron will activate these channels, which then provide an inhibitory current, damping or reducing the activity of that projection neuron for quite some time afterwards. Now, there's a bit of a caveat here, because SK channels haven't been directly observed in the hawk moth yet. But there's evidence for their existence in other insects like the fly and in mammals. And so it's possible that, that these SK channels are what is contributing to the kind of dramatic difference in effect between PTX and bicuculline application to the moth AL. All right. So mystery solved. Okay, right? So now you can think, aha, all right, so now maybe we can go back to the drawing board. We can go back to our network. We can cook up a scenario where we have essentially the same heterogeneous architecture as illustrated here, but we also endow every projection neuron with an additional, set of ion channels, which respond differently to bicuculline than they do to PTX. And that can explain this additional phenomenon. Aha, all right. Well, that by itself, so I'll explain this in a second, that's not particularly surprising, I suppose. It's maybe not even particularly satisfying, right? Because I introduced some experimental phenomenon regarding PTX, and I said, aha, okay, so we can cook up a network with the mechanism, heterogeneity, of the local neuron architecture, which can explain that particular phenomenon. And then I showed you another phenomenon which was inconsistent with my model, the bicuculline. And I said, okay, hold on. There's a new experiment here. So let me introduce a new mechanism. Aha, now I'm going to introduce SK channels to my model. And now I've patched everything up. And so now my model can explain both of these phenomena. That's not particularly, how do I say, illuminating, right? It seems like for every phenomenon I showed you, I introduced a new mechanism, and now I'm declaring victory and patting myself on the back. But wait, there's more. Because it turns out that there's additional kind of features, additional experimental observations associated with these two conditions that might actually give us a hint as to what's actually going on inside the network, and might allow us to say a little more. So the first additional phenomenon is that PTX also reduces the reliability of projection neuron responses to stimuli. That is, if you apply PTX and then you stimulate the olfactory system stimulate a glomerulus or two with pulsatile stimuli, the variance across trials of the projection neuron response increases, which isn't exactly what you would expect based on Poisson statistics. In fact, you would expect that if the projection neurons were more inhibited as a result of disinhibition after applying PTX, their mean firing rate should go down and their variance should go down. That's what Poisson statistics typically exhibit. Yet in this case, the mean goes down, but the variance goes up. So that's interesting. So what could be causing that? And the second is that when you apply BIC, not only do you see this effect, right? Projection neuron firing increases and the AHP phase decreases, but you also see that if you wait and you watch, you see that the spontaneous activity in background starts to exhibit peculiar long-term correlations in its behavior. Projection neurons in background fire rapidly for short epochs, or maybe sometimes even for long epochs, lasting 1 to 2 seconds, and then they'll be quiet for a long period of time, and then they'll fire again. So here's just a slide illustrating the experimental observation that that PTX increases PN variability without increasing the mean. And here, this is the interesting one. Well, they're both interesting for different reasons, but this is the one that I just mentioned, which is very peculiar, which is that when you apply bicuculine to the antennal lobe, you'll see there are some projection neurons and some glomeruli that fire a lot, and then a little, and then a lot, and then a little, even in background. And there are others which fire for 1 or 2 seconds very vigorously and then shut off, and then fire again and then shut off. So a question then is, what is causing this? Why would this sort of behavior manifest when you apply bicuculine? Right? And so now we have a bit of a modeling challenge on our hand, right? We have this coarse, rough idea, this coarse picture of what might be happening, but so far our coarse picture doesn't explain all of these things together, or does it? Do we need to make any more assumptions? Or is it possible that a model exhibiting, you see this? Oh yeah, I should have been doing this all along. Or a model exhibiting heterogeneous connectivity across the local neurons, as well as intrinsic calcium-dependent potassium currents within the projection neurons, maybe that actually could explain all of these phenomena. And it turns out it does. And so just to give you a flavor of the kinds of computational work that goes into this, I'll mention some of the network features, right? So we set up a model, a spiking network model of the antennal lobe, modeling several interconnected glomeruli, the macroglomerular complex, which has a couple glomeruli in it and 1 or 2 extra glomeruli, each glomeruli or each glomerulus comprising several projection neurons and local neurons. We make sure that the local neurons exhibit heterogeneity in their architecture and in what other neurons they're connected to. And we give the projection neurons calcium-dependent potassium channels so that the response of our model network to picrotoxin differs from the response of our model network to bicuculine. So in both cases, picrotoxin and bicuculine will reduce the effectiveness of inhibitory synapses, but only bicuculine will block the SK channels. This is similar to what occurs physiologically, so we'll adopt that in our model. And our goals will be to capture the phenomenon discussed so far. And here's the quick slide, the kind of slide that I said I never liked before, that I'm showing you now, right? with some cartoon depicting the model architectures, some parameters, and some equations. Now, the equations here are referred to as integrate and fire equations, which are one particular model for how any individual neuron might behave given its inputs and its internal state. There are other models of individual neuronal dynamics. There's theta neurons and Hodgkin-Huxley neurons, and the same network that I'm describing to you here also works with Hodgkin-Huxley neurons, which are maybe a more realistic model of individual neuronal activity. But I'm presenting integrate and fire equations here because they're particularly easy to understand. The way the integrate and fire equations work, and the reason I'm explaining this to you is so that I can refer to it later in a slide or two. The integrate and fire equations work by assigning every neuron a subthreshold voltage, V, which varies from a leakage potential, you can think about this as a reset potential, to some threshold potential. And when the neuron is excited, its voltage increases. When it's inhibited, its voltage decreases. And if it's excited enough, it hits threshold, and then internal mechanisms take over, and the neuron fires, sending an action potential to the downstream neurons it's connected to. After it fires, its internal mechanisms reset its voltage to the reset potential down here. And then it can continue being affected by other neurons, excited and inhibited, all right? So if you set up this model and you choose the parameters within physiological bounds, and then you tune it a little, right? Because there's a variety of different connectivity fractions and connectivity strengths. There's a relatively large range of parameters where the network actually does indeed exhibit all the phenomenon that I mentioned so far. That when stimulated by multiple pulses, the first pulse is associated with a vigorous response, and subsequent pulses are attenuated due to the AHP phase. And if you wait long enough, that attenuation is no longer seen. That picrotoxin application, the reduction of the effectiveness of inhibitory connections, actually reduces projection neuron activity due to disinhibition. And the application of picrotoxin increases the variability, so reducing the mean but increasing the variability of projection neuron responses. And when you apply bicuculine, you see an increase in activity, a decrease in the AHP phase, and more intriguingly perhaps, these long epochs in background of projection neurons firing a lot and then not firing, and then firing a lot and then not firing, with on and off phases lasting dozens of seconds. Okay, so this is just one example of projection neurons from different glomeruli firing for long periods of time. The horizontal axis down here is 80 seconds, firing vigorously in background and then not firing for a long time, and then firing vigorously again, and so forth. Okay, so in summary, right, this model that I showed here does seem to be rich enough in terms of its dynamics to allow for all of the phenomenon that I showed earlier with a single set of parameters. Okay, but why is it doing this? What's going on? And it turns out, this is what I was building up to. So I have about 5 more minutes. I just want to explain this and then I can stop. It turns out that when you set up this model and you tune it so that it exhibits the physiologically observed phenomenon that I showed earlier, the model itself ends up adopting a particular dynamic regime. A dynamic regime which is, some people call it high gain or highly sensitive or maybe balanced, where the excitatory conductances and the inhibitory conductances are both typically high and competing within the network itself. In other words, there's enough ambient excitatory and inhibitory activity, usually most of the time, that neurons are constantly in a tug of war. And if you watch what the network does, you'll see rasters that look a little bit like this. These are raster plots with the projection neurons shown in red and the local neurons shown in blue. And you'll see the activity of this network comes and goes in fits and spurts, exhibiting occasional emergent synchrony. So what is this emergent synchrony? We call these multiple firing events, which are synchronous bursts, brief synchronous bursts involving a reasonable fraction of the neurons within each glomerulus, So this is just a single glomerulus, not the whole antennal lobe. But if you look at a single glomerulus, you might see occasional synchronous bursts of this kind caused because the neurons themselves are driven close to, but not exactly up to, the firing threshold. So this is a picture of a histogram of the voltage distribution of cells within a community that exhibits these multiple firing events. The reset voltage is shown here, the threshold voltage after which neurons fire is shown up here, and the excitatory neuron distribution is shown in red on top, and the inhibitory neuron distribution is shown in blue on the bottom. And there are so many neurons bunched so close to threshold. All these neurons have their voltages just about to fire, that as soon as one excitatory neuron fires, they can drive over another, then another, then another, driving over inhibitory neurons as well, which then shut down the cascade. So these multiple firing events take place over just a few milliseconds. And because they involve many, many neurons firing in a causally connected chain, they can cause a large transient shift in the voltage distribution of the network. So this is a picture of the voltage distribution right here, right before, a few milliseconds before this multiple firing event. And then right after the multiple firing event here, the voltage distribution is shifted quite a lot. The neurons which actually participated in the firing event, shown in orange and blue, aren't that many. They're a modest fraction of the total number of neurons in the population, but the voltages of the other neurons have shifted quite considerably as a result of this volley of causally linked firing. Now, this is a kind of an idealized scenario. If we actually go into our network and look at a single glomerulus, we see something like this. Again, this is 8 seconds, this is 400 milliseconds, and this is just a zoom in of one of these little synchronous bursts, one of these multiple firing events that's only about a handful, 5 milliseconds long. Projection neurons are shown in red, and the two different types of local neurons are shown in blue and green. And what we see in this network is that the SK channels, remember the SK channels, those slow calcium-dependent potassium channels that imbued projection neurons with intrinsic inhibitory currents that prevented them from firing if they fired multiple times in response to an odor. Well, they also prevent the projection neurons from firing if they participate in too many multiple firing events in background. And so the control state of the distribution of multiple firing events is somewhat sporadic. They occur more often shortly after an input, but tend to peter out relatively quickly. But if you apply bicucoline and turn off the SK currents, then the story changes, and you can get long sequences of runaway excitation. And the runaway excitation has a couple emergent timescales, but I won't go into that. Essentially, what's happening in this network once you apply bicucoline is you turn off the brakes. You stop projection neurons from being able to inhibit themselves. So the projection neurons in a glomerulus can activate over and over and over and over and over again until, due to luck, they happen to be quiet just long enough that another glomerulus somewhere else can pick up the slack and start firing and inhibit the first glomerulus, and so forth. So I'm running out of time, so let me just pause and summarize. So ultimately, when we set up this model and we tune it to exhibit the phenomenon that I've told you about already, it seems like the model operates in a regime of high gain, which exhibits these brief synchronous bursts, which we call multiple firing events. When we look at this model, we see that the SK currents are critical for preventing runaway excitation. If you apply bicucoline to our model, we see that the runaway excitation causes epochs of firing, followed by epochs of quiescence in the different glomeruli, just as observed in the experiment. But more, we see that the epochs of quiescence are a result of excitation in another glomerulus that's inhibiting runaway excitation in another glomerulus that's inhibiting the first. So in a sense, this model has actually offered us a prediction, one that we can test in experiment. And it turns out that actually, if you go back to experiment and you test this prediction, in fact, you find that, yes, actually, when one glomerulus, when under bicuculline, when you see one glomerulus firing many, many, many times, there's another glomerulus that's quiet and vice versa. So that's actually shown back here in one of my earlier pictures, that when you see activity in one glomerulus, you see quiescence in others and vice versa. Even when you look outside the location in the antennal lobe where you applied bicuculline. So we view that as at least maybe not an entire validation, but some kind of a hint that we're on the right track. And then the other thing that I mentioned briefly, which is that application of picrotoxin increases the variability in the projection neuron behavior. Remember how I said that we expected this network to be operating in a regime that exhibits some kind of disinhibition, that there are multiple, or there's a spectrum of different local neuron types. Well, when you apply picrotoxin, due to disinhibition, more different kinds of local neurons join the party. And if you take a step back, loosely speaking, just to kind of briefly summarize what happens, the different kinds of cascades that can occur within any particular multiple firing event become more complicated when you have more neuron types at play. So under picrotoxin, you have more multi-class, multiple firing event. In other words, you have a broader zoo of different kinds of causal events that can cause neurons to fire, which thus increases the variation, not just of the projection neuron firing events, but of all the neuron firing events. So that's another hypothesis that we could test in experiment. So, so that's it. I think I'll stop here. Perhaps the mystery is solved, although there's of course more experiments to look at and more phenomenon to think about. But in summary, we believe that the mechanisms that I mentioned so far, heterogeneity and slow currents, slow intrinsic currents, can be used to produce a simple model of the moth antennal lobe that explains a host of phenomenon, the ones that I showed you so far, and which points towards a dynamically active operating point, dynamic regime, where neurons, even in the absence of input, can collude to produce these short bursts of synchrony, which we call multiple firing events, which have interesting dynamic consequences. So thank you for your time.